Welcome to the nervous system lecture. In this lecture, we're going to cover the anatomy of the nervous system briefly before moving on to the physiology of it. We're going to be covering the different subsections of the nervous system, their functions, and how it affects different organs in your body. The nervous system is made up of three regions: a sensory region, an integration center, and a motor response output. The sensory region is basically what it says. It senses changes inside and outside of your body. And it does this through receptors. As you know, you have receptors that sense external changes as well as internal changes. That sensory information will end up going towards the integration center, which is your brain and spinal cord, for processing. Your brain will decide what to do with that piece of information, and it will, as a response, send an output signal through the motor division. The motor division is made up of neurons that stimulate effector cells. And effector cells include muscles like skeletal, smooth, and cardiac muscle, as well as different glands in your body. Now, the nervous system anatomically is broken up into two major divisions: the central nervous system and the peripheral nervous system. The central nervous system includes the brain and the spinal cord. We're not really going to spend a lot of time on this. Our focus is going to be here on the peripheral nervous system. The peripheral nervous system includes every neuron outside of the brain and spinal cord. There's an afferent division of the PNS and an efferent division of the PNS. The afferent division of the PNS is your sensory input division. This is the division of neurons that travels from the PNS towards the CNS. The efferent division is the motor output division. This is the division that's exiting the CNS. So from CNS to the PNS is your efferent division. When you're sending output signals from the CNS to the PNS, you have a decision to make. If you are going to go towards the somatic region or the autonomic region. We're going to cover these two in the coming slides. So in this diagram that you see here, we've broken up the nervous system into a pathway. Make sure that you know this pathway, starting here at the receptor side. You're going to sense changes internally and externally. Externally being the somatic sensory system, internally being your visceral sensory system. And you're going to send those changes through the afferent division. Remember, afferent goes from PNS towards CNS to be integrated by the brain. Once the brain integrates that information, it sends it out through the efferent division, back out towards the PNS, and it either sends it towards the somatic nervous system, which will innervate skeletal muscle. And underneath this, put a little note that this is a voluntary system, or it will innervate the autonomic nervous system, which is basically your automatic nervous system. This is the involuntary system. Under the autonomic system, your body has another decision to make, whether it's going to go through the parasympathetic or the sympathetic regions. If it takes the path toward the parasympathetic, that means it's going to be part of your rest and digest division, essentially your relaxing division. These neurons will innervate smooth muscle, cardiac muscle, glands, and adipose tissue, but it will innervate them to respond in a relaxed fashion. If you innervate the sympathetic division, that's going to innervate the same exact regions. However, it's going to have the opposite effect to put you into a panic response. We're going to go over the different effects of all four of these tissue types in this class. In this lecture, we're going to go over the smooth, cardiac, and gland regions and how they are affected, whether you take the parasympathetic or the sympathetic divisions. Now, to send all these signals, you need a special cell type, and we call this neural tissue. And within neural tissue, you have neurons, or you have a helper cell called neuroglia. Neurons are those really long cells that you learned about in anatomy. We do have a few different types of neurons I'm going to teach you about today, but these neurons are for processing, transferring, or storing information. However, the majority of the cells in your nervous system, and this is kind of a fun fact that not a lot of people know, is the majority of those cells are the neuroglia cells. You have many more neuroglia than you do neurons. The neuroglia are your support cells. There are 6 different types of neuroglia that we're going to cover. I need you to know the name of the cell, which nervous system division it's found in, and the function of the cell. So the CNS has 4 neuroglia and the PNS has 2. We're going to start with the ones in the CNS and move on to the ones in the PNS. In the CNS, you have those 4 neuroglia, those helper cells, and you'll see all of them in this illustration, and we'll go through them one by one. The first one I want to cover are astrocytes. Aster means star in Latin. Astrocytes are cells that look like stars. As you can tell, you've got the cell body and the nucleus here with the many extensions off of that cell body, making it look kind of like a star. Astrocytes have many functions, but the first thing I want you to notice is where the extensions from this astrocyte are bound to. On this side, all the legs are bound to the axon of a neuron, while on this side, it's bound to a capillary. Remember that in the capillary, you have red blood cells, you've got plasma, nutrients like glucose, oxygen. So these astrocytes provide nutrients for the neurons. They take the nutrients from the capillary and move it into the neuron. Neurons use a lot of ATP, and you'll know this a little bit later in this class, but the ATP in neurons is required to supply energy for their sodium-potassium pumps. You learned about this particular pump in the first exam. So the astrocyte provides nutrients for the neurons, nutrients including oxygen, ATP, glucose, all the good stuff to keep the neurons working. It also is there to create a blood-brain barrier. It is there to regulate the concentration of salts and ions within the central nervous system. And if by chance you somehow damage the neurons or the tissues in the central nervous system, astrocytes are responsible for creating scar tissue. Now, the next cell we're going to cover is an oligodendrocyte. Those are these guys right over here. So these are the 2 legs of this particular oligodendrocyte. Oligodendrocytes have one big job, and that is to create myelination on the axons of the neurons. If you recall from anatomy, neurons have electrical signals running through them, and the best way to insulate them is to myelinate these axons. Myelination is just wrapping of these cells around the axon, creating an insulated barrier so that the voltage traveling through the neuron doesn't escape. So these cells are called oligodendrocytes. I want you to notice one thing about these guys, is that one oligodendrocyte can myelinate 2 different neurons. And I want you to keep that in mind, because when we talk about the peripheral nervous system, you're going to see cells that myelinate those neurons, but it's going to be one of those cells to one axon versus this one cell to 2 different axons. At the end of the day, the function doesn't change because of that. It's just a matter of understanding that there is a difference between the cells. So oligodendrocytes create myelin within the CNS. Third cell is the microglia, these little tiny guys. Microglia are small and they're mobile. They can move around the central nervous system, including your brain and spinal cord, and these guys act as the specialized immune cell of your central nervous system. They have the ability to do phagocytosis. If you guys recall from the first exam, phagocytosis is the ability of a cell to engulf and basically digest any kind of pathogen. And the last cell are these pink ones up here. These are the cells that line the central cavity of the spinal cord, called ependymal cells. This little opening here is the central cavity. These ependymal cells help to circulate CSF, that's cerebrospinal fluid, and that's the fluid that circulates throughout your brain and then down your spinal cord through this little opening in the middle. And again, that little opening is called the central canal. Then you have the peripheral nervous system. And the peripheral nervous system, lucky for you guys, only has two types of micro cells in them. These cells are called Schwann cells and satellite cells. Schwann cells are the same type of cells that we saw in the central nervous system as oligodendrocytes, but these cells create myelin sheaths by wrapping only a single axon. So each of these individual kind of node-looking structures is a Schwann cell. So there's 1, 2, 3, 4, 5 Schwann cells on the axon of this neuron. So the function of this Schwann cell is very simple. It's just to create a myelin sheath. It has the same exact purpose as those oligodendrocytes in the central nervous system, where you create a myelin sheath around an axon to insulate that electrical signal and to prevent that signal from bouncing out or diminishing by the time it gets from the beginning towards the region of the effector cells. Now, there's two ways you can create myelin sheaths in the PNS. Schwann cells are able to either wrap themselves around the core axon if the axon is very thick. However, if the axons are very thin, it's able to myelinate several different axons by almost doing a type of endocytosis, but you're not engulfing the axons into the cell, you're just creating a barrier around them, that insulation barrier. Again, function of Schwann cells and oligodendrocytes are both to myelinate the axons of neurons and to insulate the signal, helping that signal propel much more quickly and to maintain strength from beginning to the end. The last type of neuroglia we're going to cover is the second one in the PNS called satellite cells. And I'm going to use my pen here to kind of draw out what a satellite cell is. So if this is my cell body of my neuron, there's my axon, my terminal, and there's that. Let me change the color of this guy. Satellite cells sit right on top of the cell body. They are not found around the axons. And satellite cells do basically everything for the PNS neurons. They provide nutrients, they help to develop scar tissue if this cell gets damaged, and they maintain the salt gradient in the surrounding environment. Now there's a term here. Go ahead and highlight that. When you have several cell bodies in the PNS together, you call that a ganglia. So I'm going to draw, there's one neuron, there's another neuron, here's another neuron. In the PNS, the cell bodies tend to cluster together. This region of clustered cell bodies is what we call a ganglia. Think of it as a gang of cell bodies, but it's only called a ganglia in the PNS. So this just covers a little bit more in depth about myelination. And myelination, again, if you can tell here, with individual cells, these would be Schwann cells. They engulf and encircle these axons at the center. And in between the Schwann cells, you have these gaps right here and right there. Those are called nodes of Ranvier. Nodes of Ranvier are small regions of unmyelinated area between the Schwann cells. They're important because voltage is able to jump from one node of Ranvier to the next one, and that's what helps to propel the signal faster. Rather than forcing that voltage signal to run all the way through the axon, it can just bounce to get to its destination. The other thing here that's important is the fact that myelination is white in color. Schwann cells and oligodendrocytes have a lighter color than the tissues of the actual neurons. So whenever you see white matter and gray matter in brains and spinal cords, the white matter is where you have the myelination. And if you recall, we myelinate axons only, which means that the white matter is made up of axons. The gray matter, the unmyelinated region, is everything but the axon. So if you have a neuron that looks like this, you've got the axon coming down, and then you've got the terminal here. This area right here would be white matter. The cell body and this terminal would be gray matter because there's no myelin sheath in the cell body or the terminals, and therefore it's going to be gray in color. So we just covered neuroglia, the helper cells. Let's talk about neurons now. Although neurons do not make up the majority of your nervous system, we do have different types of neurons that are found. We're going to go over the structural classifications of those neurons now. And basically what that means is we're just going to talk about neurons based on their structure and how many extensions there are from the cell body. In an anaxonic neuron, here's your cell body, but you have no axon. Therefore, you call it anaxonic. We don't really know what these guys do, so you don't have to know what they do. Multipolar neurons, these ones are the most common. Again, here's your cell body. You have several extensions coming out of them, whether they're dendrites or axons. This is called a multipolar neuron because it has multiple extensions coming out of them. Bipolar neuron. Bi means two. Cell body, we have 1 and 2 extensions from it. You've got the dendrites sending an incoming signal to the cell body and then sending that signal down an axon. Bipolar neurons are specialized. They are only found in your special senses: eyes, ears, and nose. You're going to see these types of cells in the next lecture on special senses. The last one is called a unipolar. Uni means one. Here's your cell body, and there's one extension off the cell body, and it has a very, very long axon. These long axons are there to travel very long distances. Now, in terms of functional classifications, we just covered structural classifications. Now let's kind of shift gears to functional classifications. The functional classification of the neurons, you already kind of went over in the beginning of this PowerPoint. You have sensory neurons, which sense changes in the environment. Motor neurons, which send output signals to respond to changes in the environment. And then you have these guys here. Associate neurons, also called interneurons. These guys are the most common type. You have about 20 billion of them, and they are all multipolar. And their job here is to send information between neurons in the CNS. Now, these two here, sensory afferent neurons. These are found in the PNS, and they're sending signals from the PNS towards the CNS. So you sense things in the peripheral nervous system, let's say your hand, and you're going to send that sensory input to the central nervous system to be integrated. Most of these are going to be unipolar. Some of them are bipolar. Again, what did we say about bipolar neurons? They're only found in the special senses. So when you're sensing things from the eyes, the ears, places where you have specifically bipolar neurons, those guys are going to be your specialized afferent neuron that sends signals towards the CNS. Otherwise, everywhere else is unipolar. Once you go to the CNS, you integrate the signal. The CNS decides what it wants to do with that signal and sends an output response towards the motor efferent division. The motor efferent division sends signals from CNS back out to the periphery of the body. All of these are multipolar. I would like you to know what kind of neurons structurally you find in each of the different functional classifications of neurons. Now, I kind of want to piggyback on this bullet point right here. When you send a signal from the CNS out towards the PNS, that response is going to come out of what we call a terminal bouton. This is the end of an efferent neuron. It's going to send a signal out towards some sort of effector cell. It could be muscle, it could be glands. The thing coming out of this terminal bouton is going to be neurotransmitter. You're going to see more about this when we talk about the special senses and when we talk about muscle contractions. However, just to kind of touch on this a little bit, when a neuron sends a signal out towards its terminal, it's able to release these vesicles using exocytosis. So it releases signaling molecules onto the effector cells, and those signaling molecules will bind to receptors to cause some sort of change. I don't need you to know more about this just yet. I just want you to know that wherever a neuron interacts with an effector, it's called a synapse, and it releases chemicals called neurotransmitters. Neuro because it comes out of a neuron. Transmitter, it is a type of chemical that is sent on the effector cell. Now, star this slide. Why? Because the terms in blue are terms that you must know for your next exam. So I'm going to draw a line in the middle. I'm going to put the word PNS here, and I'm going to put the word CNS here, and I'm going to draw some neurons. I'm going to draw a neuron here. And remember, cell bodies all cluster together, and axons tend to cluster together. And the terminal, and terminal. I'm going to do the same thing with the CNS. Let's just draw all the cell bodies, my beautiful artistic skills. And here are the axons, the axon, and the axon. So in the PNS and CNS, you generally have your cell bodies clustering together and the axons clustering together. The regions that you have clustering have names. In the PNS, the region where you have the cell bodies clustering together, if you recall, we already covered it, it is called ganglia. So where cell bodies cluster together in PNS is called ganglia. Where the cell bodies cluster together in the CNS, it's called a nuclei or a center. However, I will not be using the word center on your exam. In the PNS, the region where the axons cluster together is called a nerve, like your sciatic nerve. The region in the CNS where the axons cluster together is called a tract or a pathway. However, again, I will not call it a pathway. I will simply call it a tract. Be sure to know these names. They're just point blank memorization questions on your next exam. So as you already know, the nervous system is broken down into two major divisions, the peripheral division and the central division. For the rest of this PowerPoint, we're going to focus on the peripheral division. The peripheral nervous system, again, that nervous system that's found outside of your brain and spinal cord, is made up of the autonomic nervous system and the somatic nervous system. Anytime you see autonomic nervous system, I want you to think automatic. This is going to be your involuntary nervous system. The somatic nervous system is the system that is connected to your skeletal muscle, and therefore, this is the voluntary nervous system. The autonomic division is broken up into the sympathetic or the parasympathetic responses. Each of these has a type of motor response that's going to require different receptors for them. The sympathetic receptors are called adrenergic receptors. These are receptors that bind to neurotransmitters like epinephrine or norepinephrine to elicit that fight or flight response. The parasympathetic receptors are called cholinergic receptors. These receptors bind to neurotransmitters like acetylcholine that help with your rest and digest response. The somatic nervous system also secretes acetylcholine, but it's specifically for the voluntary movement of skeletal muscle. So all of these are talking about the motor outputs and how we accomplish those motor outputs. So again, for autonomic nervous system, you either bind to adrenergic or cholinergic receptors, which determines what kind of response you have, or you simply have sending of acetylcholine signals to skeletal muscles. So this slide is really important. This is the pathway by which we're able to sense, integrate, and send motor output signals. So let's say we're sensing something on the skin. That sensory signal is going to go through the peripheral nervous system, and it's going to enter the spinal cord through the dorsal root and dorsal horn. That incoming signal will be integrated in the brain. We'll draw the brain right there. The output signal will go back down the spinal cord, out of the ventral horn, the ventral root, and to the effector cell. So again, the sensory afferent signal goes in through the peripheral nervous system into the central nervous system, aka the spinal cord, through the dorsal root and the dorsal horn. It'll go up to the brain to be integrated. A motor output signal will come back down the spinal cord, out of the ventral horn and ventral root, until it goes to an effector cell. The two things I want you to notice here: that the incoming signal was through a unipolar neuron. Notice here you have the cell body and a single extension from each of them. When the cell bodies cluster together, it's called a ganglion. Remember that? And then the output signal is through a multipolar neuron here. So again, the incoming signals are through a sensory afferent neuron towards the CNS. The outgoing signals are through a motor efferent neuron going out towards the PNS. Do you guys remember from anatomy? This is called the dorsal root ganglion. So I made a mnemonic to help you remember that. SAD MEV. Sensory afferent nerves go through the dorsal side of the spinal cord. Motor efferent nerves leave out of the ventral side of the spinal cord. So I'm going to go to the back picture again. SAD MEV. S-A-D. There we go. SAD. Sensory signals travel through the afferent pathway through the dorsal side of the spinal cord. Motor, efferent, ventral is MEV. Motor signals take the efferent pathway out of the ventral part of the spinal cord to the effector cell. So SAD MEV. I'm going to star this slide. Make sure you know this slide. So, types of nerve fibers. This should not be new information to you. This is basically me putting together the words somatic, sensory, and motor. Somatic always means skin or skeletal muscle. Visceral always means something internal, like internal organs that are hollow, like stomach, anything in the digestive system, reproductive systems, and urinary system. Somatic sensory and visceral sensory. I'm going to put them into one block together. These are types of sensory nerve fibers that either sense information from the somatic region or the visceral region. Somatic region includes touch, pressure, temperature, anything you can feel on your skin as part of your somatic sensory. The visceral sensory is anything you can sense internally. What do you sense inside of your body? Nausea, hunger, cramps, could be pain, could be temperature changes internally. And then the motor division is broken up again into somatic and visceral. Motor just means it sends a motor output to create a change in that region. Somatic motor, in this case, somatic has to do with skeletal muscle. In the case up here, it had to do with skin. Somatic motor is going to be a motor output that innervates skeletal muscle. Again, the voluntary system. Visceral motor is going to be an output that innervates everything else, including smooth muscle, cardiac muscle, glands. That's going to be all part of the autonomic nervous response. So visceral motor is an output signal from the brain towards smooth muscle and glands. And that output signal is also called the autonomic nervous system because it's involuntary and automatic. Do you remember what the autonomic nervous system is broken down into? Sympathetic and parasympathetic. So let's talk about that autonomic nervous system for just a moment. The autonomic nervous system is, again, the automatic system. It is not under conscious control, so we call it involuntary. And it innervates places like the cardiac muscle, smooth muscle, which is found in many different places in your viscera, and some parts of your skin, like the hair cells on them. So what exactly is the difference between somatic and autonomic? We'll go over this first, and then we'll focus on just autonomic. Not only is somatic voluntary and autonomic is involuntary, there is another major difference. The somatic division, the division that goes from your brain, that's my amazing brain here, out towards your spinal cord, and from the spinal cord out towards, let's say, your bicep muscle. The somatic division is a single neuron. It starts in the spinal cord and it goes all the way towards the muscle it's going to innervate. You can imagine these are pretty long. So somatic division has single neurons innervating them, whereas the autonomic division is made up of two different neurons. You have what's called a preganglionic neuron and a postganglionic neuron before reaching, let's say, your heart. So again, major difference: somatic, single neuron extending all the way to the muscle. Autonomic, it's a series of two neurons going towards whatever the effector cell is going to be. So here's a great illustration. You have your spinal cord here, and you have one really long neuron going towards skeletal muscle. And because it's one long neuron, I know this is part of the somatic system. When you have two neurons, so we got one here, and it's creating a synapse with a second one here at a region called a ganglia, we know this is part of the autonomic nervous system. This is true for all autonomic output signals. It is always a series of two neurons. So this picture is just coming up again just in case you want to have more examples of what's included in the sensory and then the motor divisions. Where we're going to pick up on this picture right here is right down there. Sympathetic and parasympathetic. The sympathetic nervous system is your fight or flight response. These are the major outputs that take place that I'd like you to memorize. The sympathetic response is there to help your body cope with very stressful situations. It makes sure that organ systems that are needed during panic responses are turned on and the ones that aren't needed are turned off. So we're able to do vasoconstriction. That is constricting blood vessels. Let's say the blood vessel diameter is like this. It's going to constrict them and make them smaller. And if you make the blood vessel diameter smaller, you're going to increase blood pressure, therefore increasing the flow rate. You're going to increase heart rate during the sympathetic nervous system, get that heart beating faster, more blood pumped, increase respiratory rate. Let's get more oxygen flowing through the blood. You may experience cold, sweaty palms and pupil dilation. Do you know why you have pupil dilation during your panic response? To increase your peripheral vision. These are not the only things that happen during the sympathetic nervous system response, but they are the ones that I would like you to definitely know. The parasympathetic is pretty much the opposite of that with a few additions. We're decreasing the heart rate, and you're also decreasing the respiratory rate here. You're increasing your GI movement. That means your stomach is churning, your intestines are pushing food forward in a process called peristalsis. Your urinary sphincter is relaxing, so your urinary system is functioning. You're able to excrete. And you have vasodilation. So you take your blood vessel, you dilate it a little bit to lower that blood pressure back down to normal. So there are different chemicals that'll initiate different nervous system responses. And it not only depends on the chemical that's released, but the type of receptor it binds to. If you recall a few slides back, we talked about adrenergic and cholinergic receptors. So keep that in mind as we talk about these chemicals. In the blood, you always have a certain concentration of epinephrine. This guy right here. Epinephrine is pretty constant in regulating your heart rate. It makes sure that the heart rate doesn't fall too low. It makes sure that your arteries aren't too vasodilated, so it allows them to vasoconstrict a bit. And it makes sure that blood flow is pretty even throughout your body. Now, what changes in a sympathetic response is the norepinephrine levels. Norepinephrine is generally very low in your blood. As the sympathetic response begins, norepinephrine spikes up, and that spike in norepinephrine is what really causes the changes in your heart rate, respiratory rate, and the vasoconstriction or dilation. So, tying this in to the receptors that we talked about, adrenergic receptors and cholinergic receptors. We're going to focus on the sympathetic nervous system, so we're going to focus on adrenergic receptors. Adrenergic receptors are the receptors that, when bound by norepinephrine or epinephrine, will cause the sympathetic nervous system responses in different organ systems. Norepinephrine binds to two types of adrenergic receptors, alpha and beta. There are two types of alpha receptors and two types of beta receptors. Alpha 1 and alpha 2, we'll talk about first. Let's say this is the presynaptic neuron, and this is the postsynaptic neuron. So the presynaptic neuron has the alpha 2 receptor on it. The postsynaptic neuron has the alpha 1 receptor on it. The presynaptic neuron is what actually releases the neurotransmitters out into this synapse region. And let's say we're talking about norepinephrine. The presynaptic neuron will release norepinephrine. Norepinephrine will bind to the alpha 1 receptor to cause change in whatever effector cell this is. Let's say these are cardiac muscle cells. When norepinephrine binds to alpha 1 receptors on, let's say, cardiac muscle cells, it's going to cause the heart to beat faster. Alpha 1 receptors are generally found in the smooth muscle cells of the blood vessels, the bladder, penis, uterus, and the iris. So these alpha 1 receptors are always going to be postsynaptic. Alpha 2 receptors are always going to be presynaptic. So let's draw this picture again. This is your presynaptic cell releasing norepinephrine, and norepinephrine comes and binds to the alpha 1 receptor. The alpha 1 receptor will cause some sort of downstream effect. Eventually, you will have a buildup of norepinephrine in this synaptic cleft, and it will bind back to the alpha 2 receptor to signal that that signaling cascade has completed its run. This is a type of negative feedback. When your neurotransmitter that usually binds to this neuron builds up in the synaptic cleft to the point where it binds back to the alpha 2 receptor to tell the presynaptic neuron, hey, we already sent the signal down here. Now we have a buildup of that neurotransmitter. Stop releasing more neurotransmitter. This is a type of negative feedback. The next type of receptor are the beta receptors. And the beta receptors, there's a beta 1 and a beta 2 receptor. Beta 1 receptors, these are the primary ones found in the cardiovascular system and in the stomach. In the cardiovascular system, beta 1 receptors bind to norepinephrine to increase contractility, that means the strength of the heart contraction, and they bind to the AV and SA node. So beta-1 receptors are found in the muscle, in the AV node, and in the SA node to increase strength of contraction and rate of contraction. Beta-1 receptors are also found in the stomach. When norepinephrine binds to beta-1 receptors in the stomach, it increases ghrelin secretion. Now, you don't know the function of ghrelin yet because we haven't gone over digestive system, but ghrelin's function is to make you feel hungry. So when you have norepinephrine binding to beta-1 receptors in the stomach, it's going to cause the stomach to make ghrelin. Ghrelin's going to go to your brain and tell you that you're hungry. This is oftentimes why when you're stressed out, you feel hungry. Beta2 receptors. These are the only ones we're covering that predominantly bind epinephrine. Epinephrine, remember, is constant in your blood, but when you do go through a sympathetic response, its levels do increase, and that increase is detected by beta2 receptors. Beta2 receptors are found specifically in the blood vessels of the heart, like the coronary artery, the anterior descending artery. You got the marginal arteries, if you recall from anatomy. It helps the uterus relax, and it dilates the bronchioles. You dilate the bronchioles to get more air in and out. So what I want you to know is the locations of these 4 receptors, what kind of chemical they bind to, and what happens when you bind to those receptors. So if we were in class, we would have done this practice. If you would like, you can do this on your own. This is not due, but this will definitely be helpful in understanding this PowerPoint. So there are slides that come after this. However, these are not slides that I'm going to go over with you. They are review slides. Make sure that you go over them and let me know if you have any questions.